Hi, and thank you for making it to this presentation. My name is Mehak, and I am currently a second year student at Imperial College London, studying medical biosciences with management. And today's topic, like the slide says, is going to be about human immunity. Um, I think it's one of the most fascinating systems in the human body. And so I thought this lecture series or this particular topic would be quite interesting, especially with the current situation. I think the pandemic by itself causes so much fear and uncertainty. Sometimes if you know little bit more about, how your body protects you from it, it's always useful. So we'll talk about human immunity and we'll try discussing a few things. Regardless of the current knowledge that you have on human immunity, this lesson will probably build up on it. So there's no information that you should have known prior to this lecture. I'll try to cover it all. Might as well, also introduce maybe a few new topics that you might have not heard of, which are quite interesting. So I hope this will still be interesting to you regardless of what you know about it. And we can begin. So to truly understand what human immunity means, I think we first have to explore what the word immunity by itself means. Just without the scientific definition, it really just means being protected or, being protected from something, or you're kept safe, essentially. And that's exactly what it means in the scientific basis as well. In fact, it's a state of certain resistance, or it provides a certain specific resistance to immune infections and diseases. Specifically, in the case of the human immune system, I like to break it down into the ability of our body or the system to do three things. The first thing it should be able to do is to identify, the invading microorganism or the organism that is trying to enter our bodies. A side note, immunology has a lot of key terms that you need to know to understand things, but we won't be talking much about those. But there is one key term that I'd like to focus some attention on, and that is the word I'll be using, which will be a pathogen. A pathogen is basically any kind of organism or microorganism that tries to enter your body and is not part of the cells of your own body. So that's what we'll call a pathogen. So at any point, if I shift from microorganism to pathogen, they're the same thing in this case. So the three features of the immune system should be to recognize an invading microorganism, to take an action to neutralize it or to kill it, and finally to release chemicals that will retain the memory for the next attack. This is, if you think about it, this is very similar to the system that we have in place for any kind of defense mechanism. We should know, we should be able to recognize the enemy, we should be able to take an action against the enemy, and we should remember that the next time they attack, we should have a strategy or we should have a plan in hand. So the immune system is like a bunch of little soldiers that have this plan. Since we're talking about human immunity, I think it's very important to also think about the other organisms in nature. So almost all organisms, from plants to bacteria, have inbuilt systems that prevent invading pathogens in the environment, but depending on their needs. So different species and different animals and different microorganisms have different requirements. So their systems are very different. For example, I feel like a major technology that is currently in use for gene therapy, gene editing, and it's I think definitely one of the newest features that has come up in the industry of biotechnology and even biomedicine. It's called CRISPR. It stands for cluster regularly interspaced short palindromic sequences, like it says here. The reason it's quite famous today is that it's actually used like a gene editing tool. So what it does by itself is that it cuts portions of your gene, or like not your gene, like the invading microorganism's gene, and in fact incorporates it as part of its own DNA or its own genome, which basically means it has a memory of it the next time the same pathogen invades it. So it's almost like it has resistance to it. So that is quite interesting. It's a very common example that's used in the field today. And since we're talking about human immunity, I felt like we should first start off and discuss the different types of cells, tissues, and organ systems that um, make it up. So, when we're gonna try to explore this entire system in terms of barriers. So, any kind of defense system would have like a certain barrier around it, or basically restrictions for each pathogen. So, the first I'd like to talk about is natural barriers. So, what the term natural barrier means that they keep they're not meant to keep only pathogens out. They keep everybody out. Like your skin is meant to protect you from harm, pathogens, from everything. It's just basically like a large sheet that protects your body. So these are just common features. So the three main types of natural barriers are skin, the bowel, or the insides of the stomach, and the mucosal membranes. These are basically the inner layers in our body that secrete mucus, and they're found in the nose, throat, bladder, and genitals. And we'll explore their functions later on, but these are what we call natural barriers once again. Next, when we think of immunity, we can't help but ignore what we know about white blood cells. So, white blood cells are very special in fighting all types of infections, bacteria, parasites, and this is their main role. So, where they are made and stored is essential. There are main two organs and one region in the body where they are either made, stored, or matured, and they're mainly made in the bone marrow, which is the inside of which is the in the lumen or like the inside of our large bones, and it's a soft tissue where these cells are made. And then we have two other organs called the thymus and spleen, as highlighted in this picture. So now that we know what are the different systems we have in place and what we need to do, like what does the system need to do in order to protect yourself from a pathogen, do we really know what a pathogen looks like? Like we can see, when we look into a microscope, we can see that one cell looks different than the other. So that's what a pathogen looks like. Or we know the shape of bacteria or the shape of a virus. But how do cells recognize that? Cells have very special proteins on their surface that are called markers. So these are like just little structures that surround the outsides of their cells. And this is very important. It is a very important function in helping cells realize what's a cell of their own body, like if it's a cell of my own body or if it's a foreign object. So I don't know, I'm not sure if you've heard of the concept of receptors, but it's very similar to a lock and key mechanism. So your body tries to see if that key that is on the surface of the invading pathogen matches the lock you have. So we have special receptors on the surface of our body, or not body, I mean our cell, and these keep lining up to the proteins that are present on the outside of these cells. And if they match, that means it's a cell from our own body. That's what we call a self marker. So that key, the key as I like to call it, or like that antigen or marker, is a part of that is the same type as sorry that is a marker that will be present on your own body cells as well. It's a very important feature. And anything, if the receptor does not match that antigen, those kind of antigens will have a different structure and they'll have different shape. That's why they don't match, and they will be called non-self antigens. Hi, thank you for joining. We've already started, but we're currently in the section where we talk about identifying pathogens. And if it's possible, could you please mute yourself? Thank you. But yes, you've hardly missed anything. It's just a short introduction into immunity. So, going back to what I was saying, we have these special proteins called markers on our surface, and these markers have to be complementary or they have to match the receptors our own cells have. So our own cells will have a receptor that will match with the marker that's on another cell. If it matches, we call them self markers, and they're made out of special proteins that are called MHC proteins, which is a special class of proteins found in all human vertebrates. All humans are vertebrates, so yes. Also, another thing to remember is that in immunology, often a few terms might be interchanged. So, although the second slide with identifying non-self says antigen, antigens are also understood as markers. So, if you understand the context of the sentence, you won't get confused. So, antigens or markers are basically like those structures on the exterior of the cell. If it matches with your receptor, it's called a self marker or a self antigen. If it doesn't, it's called a non-self antigen. Okay. So moving on. Now let's actually get to talking about the immune system. And I like to think of this entire system as a house. It's a very simple explanation, but it works every time, in my opinion. So you think of it as a house with gates, or front gates, or like a barrier, and a security camera. The main function of the security camera, or like even a gate, is to keep everybody out, right? Even when you enter your own house, you have to pass a gate, and everybody has to walk through it, like regardless of if you know them or you don't know them. And the camera just sees what's happening. So this is what the first part of our immune system is essentially like. It's very similar to this because it's non-specific. It's a very important key term in immunology, and it's called natural immunity. So it's meant to keep all types of objects in our environment outside. So this is our first line of defense called natural immunity, and it's or innate immunity, as you can see here in this diagram. It's called innate because it's a part of your body already. It is not triggered by anything, like in the presence of something, it's not triggered. It just sees what's happening and it's always on, essentially. So there are three main parts of this system. There's a physical barrier, there's a chemical defense, and there's basic immune responses. So if you think of a physical barrier, which is your skin, keeps everything out. And like we discussed, or at least I haven't mentioned it, but your skin has a few special features that allow it to, like, that basically gives you protection against bacteria and other microorganisms because it's a little acidic, it's a little dry, which basically is not the best environment for most microorganisms to grow in. So these are the certain physical barriers, or like even chemical barriers, like your mucus traps bacteria and pathogens, and then it's expelled by your body, which is a natural mechanism and it's always on. And the last point in the slide, which talks about basic immune responses, relates to inflammation. Now, this is when a response has to be created. So, in case, like this diagram shows you, if your skin is penetrated and if bacteria or some kind of pathogen is invading your skin, then you can see that special signals are given off, and these special cells called phagocytes move in from the blood towards the site where bacteria is collecting. And this part of the system also has a few white blood cells. There are a lot of different types of white blood cells. I don't think you need to know the specific names, but a few that are very interesting are called macrophages or phagocytes. And what it means, the word phage or phago in biology, in Latin at least, means to eat. So these cells, that's exactly what they do. They engulf, or they like capture the bacteria that is there, and it basically eats it. It, it. It's a lot complicated than that, but it surrounds it, and it basically covers the entire thing. And there are a lot of different types of cells as well. There are a few cells that release special chemicals, like the chemical signals that you see released here, are released by a few cells to alert the whole body that, you know, um, there's, sorry, there's been a penetration, and now that bacteria is in our body, we need to have a response. So these cells, they consume or they eat up the bacteria, which is called phagocytosis. Don't worry about the technical names. And that's a very important feature that we will later on talk about. But this is mainly what happens. And a few of the cells are important, but I don't think we'll go into that detail, but they're quite interesting roles in the sense that they're called natural killer cells and they're neutrophils, they're monocytes, and they're basically the first system that tries to keep the enemy or like keep pathogens away from the main body as much as possible. But in case if the pressure is too much to handle, or if the bacteria or the pathogen is stronger than we expect, we have a more stronger system in place. And I like to think of this part of the system in a different way. I like to think of it in a way that now we add like a security guard or an attendant to this in front of the house or behind the barrier or behind the front gate. So, what this person, in general, security guards have the function of making sure is like allowing specific people to come in. So, they control the movement of certain pathogens and it's specific, like they choose that this person can be allowed to enter and this person cannot be. This is very different than the first part of our system because that keeps everything out. And this feature is very unique, and that's why it takes a little longer to fight infections because it takes our time, it takes our body time to recognize these pathogens, which is the main feature here. Like our body needs to recognize these pathogens and stop them if they're not allowed. So this system, since it's non-specific, no, sorry, since it is specific and it's targeted at certain people, it's called the acquired immune system or the adaptive immune system. It means that adapting in your environment is basically changing according to your needs, right? You change according to the needs of your environment. And in this case, your body changes according to its needs. So if we just look at this part of this image and not the first two images, you can see that it's called the adaptive immune system, like we said. And it's a delayed response because it takes that much time for your body to respond to the pathogen or the antigen being in your body. And this activates the a few cells in our body that are called lymphocytes. They're a type of white blood cells, and we'll talk about them, but they play a very important role in this part of immunity. So, if you try to understand what exactly is happening in adaptive immunity, or the part where your body realizes now there's a pathogen in our system, and now we need to find a way to fight against it. And to fight against to fight against something, you need to understand that it's very important to recognize what the shape, the structure of it is, so you can create a response accordingly. So, our body has a very fascinating way of doing this in the way that the macrophage or the cell that eats organisms, like we said, in fact grabs it and surrounds it and then takes one of those antigens or the markers that we mentioned at the start and it puts it on its own surface. It almost presents it on its own surface, which basically means that your own cells in like your cells in the body can now look for that protein and now it knows the shape of that protein. So it knows, like, it's almost like an identity card. So now you know the person's name or their contact information. It's almost like that. This is a lot of information about the invading pathogen. And these cells put it on their surface and they're called antigen presenting cells. And they pass it on to two other types of cells, and that creates a whole response. But at that step, the step of presenting that antigen is very important. In fact, these cells are called antigen presenting cells because they present that part of the pathogen on their own surface. So that's what happens here. This is a little more complicated diagram, but it shows you exactly how it happens. Like they surround it and then they take it into their DNA and they put it out. You can see that this is the cell later on after it's matured. And we can see that it's presented the same protein from the pathogen or from that invading organism on its own body. Sorry. And that's when we come in contact with, like this diagram says here, a few cells called lymphocytes. Now, this whole system is so inherently focused on the function of lymphocytes, and they're, in case the infection is too strong, they have to come in to this part of immunity and they have to put up a fight. So, the different types of lymphocytes involved, there are two different types of lymphocytes. Just think of them as white blood cells that are called B lymphocytes and T lymphocytes. And there's this, if you can see, even in this diagram itself, don't worry too much about the other two T cells like CD4 and CD8, but just in the middle, you can see the B cell and there's a difference between the T cell. they have these special proteins on their surface that look very like it's similar to antigens, but it's not exactly. And that just because of the presence of these specific structures, we can understand that there's a difference in the way they function, which is very important. Sorry. Yeah. So if we try to explore the differences between the two cells, we'll understand a lot about how they function and how it helps our system. So the first thing is, like I mentioned in the second slide, or the third slide actually, we talked about the organs or the organ systems these cells are made slash found in. And so both of them are made in the bone marrow, but the reason they have different names is because of where they mature. The whole system of maturation in the human immune system, it's almost like training your soldiers. So we call these cells like the soldiers of your body, and they need to be trained. They're actually taught how to recognize pathogens in different parts. So B cells are called B cells because they mature in the bone marrow. And so basically they learn how to fight against pathogens there, while T cells mature in the thymus, which is a different part, which is a different organ, which is around your throat. So because the system is a little complicated, the different types of these cells, we'll discuss it a bit, but not in too much detail. There are two types of lymphocytes and there are three types of T lymphocytes, but their main functions are very different. The main function of B cells is to produce special chemicals called antibodies. These are what you need. That's the main basis of this whole system. If the pathogen is too strong, we need to release antibodies that will have different strategies to destroy the pathogen. And it's very important for it to be released. So those are produced by B cells. And T cells have lots of different functions. It can be to help B cells, it can be to kill pathogens, and can also, identify different cells, like, identify pathogens and alert the whole system that there are pathogens here. But the main point that B cells, the main function of B cells in this case is to actually, B and T cells, sorry, is to produce these special types of cells called memory cells. And their B cells have a memory type called B memory cells, and T cells have a type called T memory cells. It's a little confusing and it's a lot of words in that one sentence, but both of them have types of memory cells that basically have an important function that we'll discuss later on. Yeah, so now that we've talked about all of this, we said that macrophages present a part of the antigen on their surface, and they come in contact with T lymphocytes. We didn't mention which lymphocytes, but that's actually what happens. So the cells that present those antigens, APCs, like you can see here, they present it to T lymphocytes, and T lymphocytes then start this whole process. T lymphocytes alert B lymphocytes. So this goes on here. They alert B lymphocytes, and they even alert effector T cells. And this whole process mainly helps with, like we said, killing the cells. And since T lymphocytes have alerted B lymphocytes, they now produce special types of plasma cells, which are basically the cells that produce these special antibodies. And those are the antibodies we actually saw on the surface of the B cell, if you remember. You can see these are what we call antibodies, and they have this on their surface because that's exactly what you need to collect pathogens and destroy them. So B cells produce antibodies, and you can find them on their surfaces. And although T lymphocytes have these special structures, they're not actually antibodies, they're just antigens, like the ones we mentioned to recognize pathogens. So they have those to recognize, like, the key lock mechanism. It's like a receptor to recognize other pathogens. So I realize how this slide has a lot of information, but it just talks about all the cells that are involved in the system and how these systems overlap. Like we said, this was innate immunity and this was adaptive or acquired immunity. You can see that there are common cells in between the two. The interaction of two cells is what connects the two systems. APCs are actually, you can see that this cell is from this diagram, which is part of innate immunity. This connects to T lymphocytes. And the fact that these two systems like they work together is very important. And actually, that's where the role of the spleen comes in. Like we mentioned the thymus and the bone marrow, but we haven't mentioned the spleen yet. The spleen is the region where uh, immature T lymphocytes, or they're not fully mature yet, they meet these antigen presenting cells and then they recognize that this is the pathogen in the system and then they know what type of approach to go ahead with. So we've covered most of the system, and there are obviously a lot more details into how exactly it goes through. But this basic understanding is what is very important in creating vaccines in today's world. So, sorry, yeah. Um, and vaccines are based on a very important concept called immunological memory. It sounds very complicated, but it's extremely simple. And the concept is that we've encountered a pathogen or an enemy, as we like to call it, and we fought against it. And let's say now the pathogen is gone from your body has fought with it. But how do you remember, you know, um, how do you remember that the next time if this pathogen affects me, what do I do? What kind of strategy do I have? And that's a very important feature of the human immune system. So I like to think this graph explains it really nicely. So the second part of immunity that we call adaptive immunity or acquired immunity, we mentioned T lymphocytes and B lymphocytes that have special cells called memory cells. And these memory cells are basically cells that always, there are a few cells, most cells die out. Like these cells, because they're in battle, they they themselves endure some injuries, or the cell bursts, or the cell dies. So that's actually why we have, the time. If you realize if you're sick, that's your body responding to it. Your cells are dying. There's inflammation. You're not feeling too well. Your cells are dying, but a small portion of it, after you've recovered, always stay in your body. So these are memory cells that remain in your body for years, and they're very useful the second time your body faces that pathogen again. So let's just try to see a graph in which this is the time on the x-axis, and this is plasma antibody concentration, and that's basically the concentration of antibodies that is produced by your body. So initially, on like the first day you get infected, or the first time you get exposed to this antigen, your body has no antibodies because it hasn't responded to it yet. We said that innate immunity, it takes a few days, but the second part of immunity takes a lot. They take at least a few more days than the first part to fight against this antigen or pathogen. So the first time we come in contact with the antigen or the pathogen, we have like your body takes some time to build antigens, to build antibodies and to basically build a response against it. But you can see that this is slowly rising, and then it eventually reaches like a peak. So when it reaches this peak is basically when your body starts the adaptive immunity part of your system, and it releases antibodies. So after that, you fought against the pathogen, your body is recovered, then the number of antibodies slowly go down because they've fought against the pathogen. But you can see the second time, maybe you caught it again, the same pathogen or the same virus from a friend. You can see that your immunity or the plasma concentration, the antibody concentration goes high up. It shoots much faster. If you look at the steepness of the curve, it's much steeper, which basically means that since it's steeper, your body responds to it so much more quickly. It takes the same time span, but you can see that even the amount of antibodies that are produced are much higher. Like you can see that initially you've only produced this much antibodies in the first time, but the second time you were exposed to it, you got almost three times or almost four times the number of um, antibody concentration. So your body has recovered and it has fought off the pathogen the second time much more successfully and much more quickly. And this is a very important feature because your it's very important to realize that fighting against pathogens or any kind of microorganism takes a lot of energy. That's why you feel lethargic, tired, and you're not in the mood to do anything the first time, you get sick or you come in contact with a virus or any kind of pathogen. But the second time your body comes in contact with it, it can use that energy to produce a lot of antibodies. So its response is much stronger and more effective. And that's the main understanding of this system. Um, so, yeah, and this is the most important feature that scientists use. Like, based on this feature, they've built vaccines. Because if we look at vaccines, if we try to understand what exactly they are, they're basically just weakened versions of the uh, virus or the pathogen, which basically means they take that microorganism or the pathogen, they use certain parts of it, or like they restructure it in such a way where it doesn't affect your body in such a impactful way. Like your body often gets overwhelmed with infections, and that's why you fall sick. But a vaccine is supposed to trigger a response in your body without making the individual feel overwhelmed. So it gives you immunity without making you fully experience the condition. So you get a small part, or they engineer a small part of the pathogen and they insert it into your system, and your body produces the same response we talked about. It recognizes it, it produces antibodies, and then it mainly produces memory, or it remembers that it produces immunological memory against it. So the next time you get infected with it, your body will recover much more quickly. So that's the main concept of immunity. And it's very important to understand that although we have this system in place and immunity works very well, sorry, by its own, it's very complex and concise system. Sometimes there are a lot of individuals whose bodies are not able to recognize their own cells, which is which is extremely hard. It takes a toll on your body because obviously um, there are a lot of conditions that can be caused because of it. I like to use these cartoons because they perfectly show some of the conditions you face. And so the first thing, this cartoon talks about autoimmune disorders. Autoimmune disorders basically talk about autoimmune. The word itself means auto or against your own body, so against your own immune system. So it basically means your own cells think that other cells in your body are intruders or are foreign, and then they don't even recognize that they're their own cells, in fact. So your body basically attacks itself, or your immune system attacks your own body cells. And the same happens here. The story talks about the same thing. Your body recognizes it as a pathogen, or it recognizes it as a bacteria, vaccine, for whatever, uh, virus, for whatever reason, and then they send like the command to attack them. So if you like to think of, if you look at the immune system or like normal immune function, it's this cartoon summarizes it quite nicely. So if you think about it, your immune system is like a guard dog, like we said, that protects your body against threats. And there are different things that can happen depending on how severe your case is. So if you have what we call hypersensitivity disorders, sometimes it might overreact and attack other things in your body, which basically means um, this is basically what allergies look like if you try to understand that. The different serious, some people have more strong allergies, some people don't. So there's a certain level to it. But allergies basically mean that your body doesn't recognize new substance that has come in, and it attacks against it. And so it might overreact and cause more inflammation. That's an example of sensitivity, sorry. And then the next thing that can happen is immunodeficiency, which, if you just break down the word, deficiency means lack of. So it means lack of immune system, which basically means it's not functioning as well. Like before, we said it was too reactive. It recognized it was overreacting almost. But immunodeficiency means that it does not react as much. It does not give a response, and it's not responding as well to it. So it stops working and your body has no defense system up. That's why you become more prone to other kinds of conditions. Like an example of this would be AIDS, which is acquired immunodeficiency syndrome, which basically means that your body's immune system is compromised. So you get affected by even small conditions like a cold, um, common colds, flus, and these kind of things affect your body more greatly because your system does not defend against it. And um, the last kind of example in which your immune system is negatively affected is called autoimmune disorders, which basically, like the two cartoons that we've mentioned here, it talks about how your body can't recognize the difference between your own cells and foreign cells. So it thinks that your own body cells are actually foreign cells and it attacks those cells, which is basically a dog attacking its owner. It fails to recognize its own self. So that's that. And the last slide, or the last thing I want to talk about for today's whole session is something that's very new that's coming up in this part of immunotherapy, which basically covers the thing of so different patients, like you can see these three conditions here. We said that there's a different level of seriousness to it. And some people get more affected more seriously, some people don't get affected as seriously. And that's a big problem in the medical community today. The fact that medicines don't affect two people who have the same condition in the same way is a problem. That means if I treat my patient in a certain way, but they don't respond how I think they should respond, that can endanger their life even more. So we can't predict how their body will respond to it, although we feel like it should be the same. Our bodies are so different. So that means our immune systems are so different. And if a treatment is not bad, but it's not even effective, like if a treatment doesn't help my patient, it still makes them lose time, which basically puts their life in more danger, if that makes sense. So we've um, there's a big term which is called personalized medicine that has come up recently in the last 10 years, I think. And it basically wants to cover um, it's like specializing treatment for each patient. Since we know all our bodies are so different, we need to have a special system in place for each of them. It's very expensive and the technology is very new, but there's a lot of different parts to it, which means that we look at a person's body, we understand their particular system, and then we find a treatment that works for them. So this this new technology that I want to talk about is called organ on chip development, which basically covers a lot of different aspects of drug testing because immunotherapy is still very relatively new and like finding solutions for different types of um, immune diseases takes a lot of effort and it takes years of drug testing trials and so much work by scientists even on one type of condition. And often it's very expensive to do these kind of trials and examinations on animals because animals their bodies are so different than ours. Like yes, it's still very similar compared to any other species, but their bodies are very different, and it's hard to understand how a drug will react in their body and in our body. So what this system or what this technology tries to do, it tries to combine microelectronics and biology and to produce basically like a chip. This chip has a lot of different components that basically make it like an organ. It's not exactly like an organ, but it also means that um, different parts of like different tissues can come together in the same space. In fact, a small space. This is engineered together such that you can maybe even insert, like you can see here in this first example, in this first picture. You can see that there's not a picture exactly of this chip, but it looks very similar to this chip is because we're talking about the lungs here, right? So it looks at a particular organ. In this case, we're looking at the lung. Then it looks at, its basic components. We can see that this is the inner layers of what um, the alveolar sac, which is basically the smallest unit of our it's like the small air sacs we have in our lungs. And so it looks at the different layers of cells and then it reproduces those layers of cells. So it's like literally creating a microscopic, not microscopic, a miniature version of an organ. So it's compact. It has all those different layers. They have these epithelial cells, like we can see here. Then we can see epithelial cells here. We see endothelial cells. We see the same in the chip here. So it basically tries to create a small version of common organs, and then they can even maybe insert the patient's blood, like so they can use specific samples for each patient, and then check to see how a drug works, inject it into the system, and then see how the body responds, and a lot of different parts of it come into it. But it basically tries to see the response of that particular body, that particular person's blood to that drug. It gives it the perfect environment, it puts that sample in, and it tries to understand what exactly is happening. And I think obviously there's a lot of research that has to be done specifically on the function of this chip, how effective it is, and is it really simplifying drug testing or is it just not giving us the exact answer we want? Is it telling us that something's actually happening or is it not accurate enough yet? So it's very futuristic and fresh, but um, it seems like a very good representation of actual organs. So it's also great, to be honest, for all the news that has come up on testing on animals, the ethics of it, the expenses, the toll it takes on their bodies. So it's very important to acknowledge that and also try to create a special part of it that works specifically on special patients because some patients just have more severe conditions and it's very hard to tackle that usually. Um, so this is the end of the presentation. And I hope that this has made sense. Obviously, it was quite rushed because there are so many different parts to human immunity, especially the section of adaptive immunity, like we talk about. Those cells work in very specific ways and they send out special signals and very unique chemicals. So, talking about all of that is quite complicated. But if you do have any questions, please go ahead and ask them about anything for that matter. Maybe you want me to cover the introduction that you probably missed. I don't mind doing that as well. But you didn't miss much. It was just talking about the immune system and its basic functions. So if you have any questions, please switch on your microphone if you'd like to, or you can type them in the chat section. But I'll be here for any questions.
Good morning. I have
Hi.
one question.
Yeah.
Can you show me, please, the first slide or
Yes,
about
definitely.
the introduction?
Sure. I'll just quickly go through that. So this was the title slide. And just to give you a small introduction, my name is Mehak, and I am currently a second year undergraduate student at Imperial College London, studying medical biosciences with management. And as you as you heard, we covered just a brief, I guess, a brief lecture on the entire of human immunity. And so we started off with, understanding the basics of immunity. Like if we need to understand the scientific meaning of a word, we need to understand what it means just in plain English. Like what does the word immunity by itself mean? And in this case, it means um, having specific resistance or basically being protected from something. That's like the simple meaning of the word immunity by itself. But in our body, it means to give us resistance or to basically equip us with all the, it's like building a protection, essentially. It sounds a little more, I think I'm making it a little more complicated, but it basically wants to protect you from different types of infections and diseases. So you have a system in place. And the three most important parts of the system refer to recognizing and invading microorganism, taking an action to kill or neutralize it, and finally, you need to retain memory of the event for the next attack. So, in case you, like we said, in case you come in contact with the pathogen once again, your body should have like a specific plan so that it can fight against it efficiently. And although human immunity is quite interesting, so many other organisms in nature have these systems, like immune systems, that protect them from invading pathogens, depending on their needs. So I feel like this is something you might have heard of. I'm not sure, but it's called CRISPR, which is a new technology that's come up in gene editing and gene therapy, which basically it it's a technology that's used to cut small sections of your gene and to either replace them or to remove them. And that's the same. This technology comes from bacteria. Lots of bacteria have these system in their place, like in place. So when they come in contact with a pathogen, by the way, is any kind of invading microorganism, which is basically anything that's not a part of your own body. So it's not a part of your own body, and it will use your body's resources to grow, which is something we don't want. So, sorry. In this case, we call those microorganisms pathogens. So, in the case of bacteria, when they come in contact with a pathogen, what happens is that they fight against it, and they break its barrier, and they get access to its DNA, or well, its genetic code. They get access to its genetic code. They cut a small section of it, and then they include it in their own genome. Genome is like your entire set of genes. So what that basically does is that it keeps a part of their identity in your own genes. So in case you come in contact with them once again, you have a resistance against them. You know what exactly the pathogen is, so you can fight against it in a specific way, and it's quite unique and it's quite interesting as well. So we talked about that, and then we covered a bit of what is the human immune system even made of? Like, what are the different organs and cells and mechanisms that make up this entire system? So that's um, you can think of the first few natural barriers, like we said. So this can be the skin, the bowel, or the mucosal membranes, which is basically these are normal barriers that keep everything like they're natural barriers. What the word natural here means that regardless of if there's a pathogen or if there's no pathogen, these systems are always there. But if you think about it, why we focus on the word natural so much is because the second part of the system, like we talked about, the adaptive immunity, that makes a specific target plan against these like it produces antibodies and those things. Those things don't happen all the time in our body. We're not constantly producing antibodies. We don't always um, uh, activate our white blood cells, but these natural barriers are very important to keep us safe all the time. So we call them natural barriers. But apart from that, we have to consider the role of white blood cells, because we know that white blood cells play a major role in protecting us against infections and diseases of all types. And so they're produced in specific organs. So you can see that there are two organs here, and one, just a general thing called the bone marrow. And the bone marrow is like a type of tissue that's found inside your bones. So this is where all types of cells are made, like including your red blood cells, your white blood cells, all types. These are where our stem cells are stored, which is basically what renews all our other cells. Um, so all types of white blood cells or lymphocytes are made up in the bone marrow. And the spleen and thymus are mainly, like we said, used for B cells and T cells. So B cells and T cells, both of them are made in the bone marrow. But in the thymus, you have T cells, and in the bone marrow, you have B cells. Like that's where they're made. But they mature in different regions. Sorry, um, the T cells mature in the thymus. Mature is, I think I mentioned this, might as well go over it again, which is basically like um, maturing in general. When you mature in life, is because when you become more independent. That's literally what the word mature means, and that's actually what these cells go through as well. They learn to become independent, and they learn to fight against pathogens by themselves. So they train to recognize pathogens in the system. And so T cells do that in the thymus, which is around here, and B cells mature in the bone marrow, which is basically in the largest. It's not all your bones, but it's also mainly in your biggest bone, like your femur and even your hips, like the hip bone. That's where they're found. The bone marrow is found. So we talked about the different parts of the immune system that include them. And then we talked about identifying cells. Like, yes, we know that when we look into a microscope, we can say that this looks different than this. This is a cell of my own body. This has a different shape. Like, you can see just looking at this image that this cell looks very different than this cell. But how do our own cells recognize each other? Like, how do they know that this is a cell from my own body, or like from the same body, and this is a cell, this is an invading pathogen, or this is a foreign object? So we have special markers or special shapes of proteins that are found on our surface, like these protruding structures. And in humans, these types of proteins are called MHC proteins. It's a little more complex than you need to know, but it's a special type of proteins that are only found in humans. And if if a cell has that, and it's like if two cells have the same type of MHC protein, they're from the same body, and those are called self antigens because they belong to me. Like even between two people, they'll have different antigens. Like even if two people who are related to each other, their cells will have different structures on the outside of the cell. So your body recognizes them using a lock and key mechanism, which is basically like if you've heard of receptors in enzymes, like the substrate fits with the enzyme, which is the same logic here. We have receptors on our surface, and they join with the markers. They check. That's how they check. It's like a lock. You put a key into a lock. So there are different keys. You check which key matches. And if it matches with what your own body's receptors have, that means it's a self antigen. And if it doesn't match, it's a non self antigen. In this case, they don't show that mechanism, but I think this diagram might help. Like you can see how it matches here. Like it closes in. So if it closes in, then it matches, then it's a cell that belongs to your body. It's not a foreign cell. So yeah, that's mostly I think all that you missed. But I hope that's useful.
Okay, thank you very much.
Thank you. I guess this is also the end of the session. So thank you for attending. I hope you enjoyed it, and
Yeah,
yes, take care.
it was very interesting.
Thank you. Thank you. See you. Bye.
Okay, thank you. Goodbye.
Thank you. Bye.